All right, guys, so here we are in our next section. We are in chapter 9, so we're going to go through 9.1 through 9.6. It's called Frontiers of Biotechnology. We're going to be talking about some of the vocabulary associated with this section, and then we'll be watching some review videos this week and just looking at the vocabulary basically for this section. So our key concepts. Biotechnology relies on cutting DNA at specific places. The polymerase chain reaction rapidly copies segments of DNA. DNA fingerprints identify people at the molecular level. DNA sequences of organisms can be changed. Entire genomes are sequenced, studied, and compared. And genetics provides a basis for new medical treatments. We have a bunch of new vocabulary. We have this thing called restriction enzyme, gel electrophoresis, restriction map, polymerase chain reaction, also known as PCR, DNA fingerprinting, clone, genetic engineering, recombinant DNA, plasmid, transgenic, genomics, gene sequencing, human genome project, bioinformatics, and gene therapy. That's a mouthful, sorry guys. All right, our essential questions. What techniques are used to study human DNA? Why would DNA fingerprinting be important? How and why do scientists manipulate DNA in living cells? And lastly, what are some ethical implications involving biotechnology? So we need to talk a little bit about some of the techniques that scientists use. So the scientists actually use several techniques to manipulate or to change DNA. They use things like computers, chemicals, bacteria. These are all used to work with DNA. And we use these tools to do genetic research and do some different things in biotechnology. Biotechnology is just technology involving biology. Restriction enzymes cut DNA. So restriction enzymes are these special enzymes that act kind of like scissors. And the scissors are molecular scissors. They snip the DNA in specialized places. So they allow scientists to more easily study and to manipulate. To manipulate means to change genes. It cuts out DNA at specific nucleotide sequences called restriction sites. And these different restrictions enzymes can cut DNA in different ways. The main thing that you need to know is just that first part. Restriction enzymes act as molecular scissors, and it actually cuts the DNA, allowing scientists to then just look at a specific part. So there we are. And you can see in the first enzyme, it cut it into 3 spots so that you have 4 little pieces. Restriction enzyme number 2 cut DNA in many more spots, so you have several different little pieces that can be looked at more closely. Now, the restriction maps show lengths of DNA fragments. So we actually use this stuff called gel electrophoresis. It actually separates the DNA by its size. So the little pieces that got snipped or cut up into little pieces, it gets separated, it gets pulled through this gel using electric currents, okay? And it separates them by size. The smaller the pieces, the further they move through the gel. The larger the pieces, the further down they stay. Okay, and the fragments of different sizes appear as little stripes or bands in that gel. So there's a picture there. The further, again, they move, the smaller the pieces are. Okay, if they only move a little bit, the bigger the pieces are. So gel electrophoresis is used to separate DNA fragments by size. And again, you just need to know that top part. Okay, and again, same thing here. Only copy the highlighted. A restriction map shows the lengths of the DNA fragments and is useful in genetic engineering and studying mutations, so changes in DNA. Okay. The next thing we have is PCR. PCR stands for polymerase chain reaction. Now, scientists use this a lot when they want to copy a specific DNA sequence. It makes many, many, many copies of a sequence. It's similar to the process of DNA replication. And each PCR cycle actually doubles the number of DNA molecules. So basically they're copying it and copying it and copying it and copying it. Okay, so it makes many copies of a specific DNA sequence. So you want a little tiny piece and just copy that piece, you can do that. A DNA fingerprint is a type of restriction map. And DNA fingerprints are based on parts of individuals' DNA that can be used for identification. Okay? And it's basically pairs that are repeated many, many times in a DNA sample. We use DNA fingerprinting in several ways for evidence in criminal cases, paternity tests, immigration, studying biodiversity. And if you look here, I've got one sample. So this would be used in a criminal case. At our crime scene, we identified some genetic markers of our criminal. Okay, now we have our suspects, and we collected DNA samples from these suspects, and we can use this DNA fingerprint to match our suspects to the DNA found at that crime scene. Also, we can use it for tracking genetically modified crops to see if they're spreading where we don't want them to go. Another thing that scientists can do is clone individual organisms. So a clone is a genetic identical copy of a gene or of an organism. Now, cloning does occur in nature. We did talk about asexual reproduction, and asexual reproduction, like if you have budding or binary fission where one bacteria splits into two and so on, that is cloning. They're getting exact copies of the DNA. Now, cloning does have some potential benefits, like perhaps getting organs for human transplants, saving endangered animals, but it also does bring up a lot of concerns, like are the clones imperfect? Are they less healthy than the original animals? If we have too many clones, it's going to reduce biodiversity, a whole bunch of different situations. New genes can also be added to an organism's DNA. We call that genetic engineering. So we change an organism's DNA to give it new traits. When we change that, we take DNA from one organism and we implant it into another organism. So genetic engineering is based on the use of this thing called recombinant DNA. And recombinant DNA contains genes from more than one organism. So basically, we use this thing called a plasmid, which is bacterial DNA. And we use it to hold a piece of whatever we want to put into our new organism. So we put the foreign DNA on it. So bacterial plasmids are often used to make recombinant DNA. Plasmids are just loops of DNA in bacteria. We use those restriction enzymes that we talked about at the beginning that are like the scissors to cut that little plasmid, that bacteria DNA, and then we stick on that foreign DNA right into our bacterial DNA. And you can see here it is, plasmid. Okay. Then we take that, the bacteria reproduce. We take that new plasmid and we insert that into our new organism. So genetic engineering produces organisms with new traits. Organisms that have those different genes from something else we call transgenic. So transgenic organism has one or more genes from another organism inserted into its genome. So here it is. Here's our plasmid. Foreign DNA, okay, contains the genes. Then we insert that into our brand new organism to make it a transgenic organism. Transgenic bacteria can be used to produce human proteins. So we use genes. We insert in, like I just told you, genes inserted in the plasmid. The plasmid is inserted in the bacteria. The bacteria express the gene, and then we implant it in whatever we want. Transgenic plants are very common in agriculture. We call them GMOs. You've all heard of them, genetically modified organisms. Okay? And so a lot of our crops are now genetically modified. So we call them GMOs. And if you look, I would say a ton of them to make them stronger, pest resistant, contain more vitamins, a ton of different things. Okay. Transgenic animals are actually used to study diseases and gene functions. So here are some mice. They've been genetically modified, transgenic animals, and they're used to study development and disease and kind of simulate some of the diseases that we would see in humans. Now, scientists do have concerns about using genetic engineering because there may be some long-term health effects of eating genetically modified foods that we're not aware of. They might have some implications in the ecosystems on biodiversity and other stuff. So there are some problems associated with that. Genomics involves the study of genes, gene functions, and entire genomes. So genomics is the study of genomes. And we talked about genomes is all of the DNA an organism has. And it can include the sequencing of the genome. And we can use this to compare genomes within a species and then across different species. And gene sequencing is determining the order of DNA nucleotides in genes or in genomes. And the genomes of several different organisms have been sequenced. And you can actually look at how many different bases and how big the genomes of some different organisms here are over here. Now, the Human Genome Project sequenced all of the DNA base pairs of human chromosomes. It's analyzed DNA from a few people. We're still working on identifying like the maps, like what do all of the genes do and how do they interact and all sorts of things. But this was a huge project undertaken probably around 2000. It was going on since the mid 90s into the early 2000s. And technology allows the study and comparison of both genes and proteins. So bioinformatics is the use of computer databases to organize and analyze biological data. Now, DNA microarrays are used to study the expression of many genes at once. The big takeaway is, for us, we really just need to know that bioinformatics is the use of computer databases to help to organize our genetic information. Now, we can do these things called genetic screening, and you guys are probably aware of this, but genetic screening involves the testing of DNA. We do this to determine if you've got a risk of having a disorder or passing on a genetic disorder. So we detect specific genes or proteins, and it can also detect some genes that might be related to an increased risk of cancer. Now, these things, although, like we'll get into this when we watch some of the movies, although you may have a gene for it, it doesn't necessarily mean that you will express that trait. Okay, that also comes into play in your environment, and we talked about that in previous videos. Okay, what they're working on now, gene therapy. Gene therapy is the replacement of faulty genes. So what it actually does is it replaces a defective or missing gene, or sometimes even adds genes to help treat a disease. Now, we have several experimental techniques for gene therapy. Sometimes they use viruses to infect a cell and insert DNA, and to stimulate the immune system to actually attack cancer cells or kill off cancer cells. They also insert genes into correct cells to fix problems maybe they've seen. So they control which genes are expressed, which ones are turned on, turned off. There's a whole bunch of different situations that they can use gene therapy for. And again, we're going to watch some videos talking about these different things. But this is basically it for our vocabulary for biotechnology. And again, we'll watch some videos that kind of clarify some of these things.